Place your palm flat against the nearest wooden surface right now. A desk, a door frame, a floorboard. Hold it there. Notice that it does not feel cold. Notice that your hand stays warm. That sensation, that specific thermal neutrality, is the first mechanism already operating on your nervous system, and understanding exactly why it happens will change how you read every piece of research that follows. Wood has a thermal conductivity of roughly 0.12 watts per meter per Kelvin. Steel conducts heat at 50 watts per meter per Kelvin, more than 400 times faster. When your hand contacts steel, thermal energy moves out of your skin rapidly. The temperature receptors in your dermis fire hard, and your brain registers cold. When your hand contacts wood, heat transfer is so slow that your skin temperature barely shifts. Your thermoreceptors, free nerve endings clustered at roughly 1 to 2 millimeters depth in your epidermis, detect almost no change. Your somatosensory cortex, the strip of tissue running across the top of your skull that processes all tactile and thermal signals, receives data that reads, "This surface is giving me warmth." But the surface is giving you nothing. It is simply conducting so slowly that your nervous system cannot distinguish it from warmth. Every time you have held a wooden handle, leaned against a wooden wall, or felt comfortable touching a tree, you are experiencing a thermal illusion generated entirely by conductivity coefficients, not by anything the wood was offering. That is mechanism one, already done, already running. Now hold your position, because something else started the moment you entered proximity to that wooden surface, something that operates entirely through the air between your skin and the material, and it has been altering your immune system in ways that most people walking through a park never consider. Trees and the wood they produce release volatile organic compounds continuously. The word volatile here is a precise physical term, not a casual one. A volatile compound is a molecule whose vapor pressure at room temperature is high enough to allow significant evaporation into the surrounding atmosphere. Vapor pressure is the pressure exerted by a gas in equilibrium with its liquid phase. When that pressure exceeds atmospheric conditions at ordinary temperatures, molecules escape the surface into the gas phase and diffuse outward. Alpha-pinene, beta-pinene, limonene, and camphene are the dominant molecules in this class released by conifers and many hardwoods. Their molecular weights sit between 120 and 160 grams per mole, light enough to become airborne, stable enough to persist in the atmosphere for hours before oxidizing. These compounds have a collective name that carries more history than most people realize: phytoncides. The term was introduced in 1928 by the Soviet biologist Boris Tolkien, who derived it from the Greek for plant and the Latin for exterminate. Tolkien documented that these airborne molecules inhibited bacterial and fungal growth in laboratory assays. The tree was not producing aromatics for your benefit, it was prosecuting a continuous chemical defense operation against pathogens, and you were walking into the diffusion field of a biological weapons system. The physics of that diffusion follows Fick's law. Molecules move from regions of high concentration to regions of low concentration at a rate proportional to the concentration gradient and inversely proportional to distance. The bark surface is the point of maximum concentration. As distance increases, concentration falls off predictably. 1 hectare of mixed conifer forest, roughly 2.5 acres, releases approximately 5 kilograms of these volatile compounds into the surrounding air every single day. 5 kilograms. That is not a trace. That is a continuous chemical output comparable in mass to a bag of flour evaporating into the atmosphere above 2.5 acres of forest every 24 hours. The air inside that diffusion field contains 200 to 300 bacterial cells per cubic meter. Urban air outside the diffusion field contains orders of magnitude more. The antimicrobial plume is measurably sterilizing the atmosphere it occupies. When you stand near a tree, you are inside that plume. When you breathe at rest, your lungs process 6 to 8 liters of air per minute. Each breath pulls these molecules across the alveolar membrane, a surface roughly the area of a tennis court folded into your chest and into your bloodstream. Now here is the measurement that matters. In 2009, Dr. Qing Li at Nippon Medical School in Tokyo conducted a series of controlled exposure studies in which human participants were taken into cedar forests and had their blood drawn at intervals over the following weeks. The specific target of analysis was natural killer cell activity. Natural killer cells are a class of lymphocyte, white blood cells that circulate in your blood and lymph, whose primary function is to identify and destroy cells displaying abnormal surface proteins, the surface signature of viral infection and malignant transformation. These are not passive components, they are the frontline surveillance system that your body runs continuously to suppress tumors and viral spread. Dr. Lee's team found that after a single day of forest exposure, natural killer cell activity increased significantly, and that increase persisted for more than 7 days. 7 days of measurably elevated immune surveillance from one afternoon among trees. In subjects who spent 2 or 3 consecutive days in forest environments, the elevation in natural killer cell activity persisted for more than 30 days. 30 days of altered immune function from 48 hours of breathing air inside a phytoncide diffusion field. The same research measured urinary adrenaline and noradrenaline, the catecholamine hormones that your adrenal glands release during stress activation of the sympathetic nervous system. Both were significantly reduced after forest exposure. The stress response chemistry that raises your heart rate, tightens your vasculature, and suppresses digestion was measurably dampened by the same molecular mechanism that was elevating your immune surveillance. One compound class doing two things simultaneously: upregulating cellular defense while downregulating sympathetic activation. To put a number into proportion, pharmaceutical natural killer cell activators used in oncology support cost between $200 and $800 per treatment course, and they operate through overlapping receptor pathways to the ones phytoncides activate for free through a concentration gradient in open air. That is not mysticism. That is receptor pharmacology operating through gas phase diffusion, no prescription required. The evidence for the natural killer cell finding is strong and replicated across multiple cohorts. The anti-anxiety effect is well supported by the catecholamine data. The specific molecular receptor mechanism linking phytoncides to NK cell upregulation is biologically plausible and consistent with known immunology, but has not been mapped with the precision that would satisfy a phase 3 clinical trial. That is the honest evidentiary position. Strongly evidenced effect, partially characterized mechanism. Now consider the second mechanism operating through a different physical pathway entirely. The tree you are touching, if it is growing in soil, has a root system in electrical contact with the earth. The Earth's surface carries a net negative charge, maintained continuously by the global atmospheric electrical circuit. At any moment, approximately 2,000 thunderstorms are active around the planet. Lightning strikes discharge energy into the ionosphere. That charge gradient between the ionosphere and the surface drives a steady downward current of roughly 1,000 amperes globally, which deposits negative charge at the surface. The surface of the Earth is a reservoir of electrons. A tree with roots in that soil is a grounded biological conductor. When your skin, which is also electrically conductive, contacts the bark, a charge pathway exists. Ground to root to trunk to bark to skin. Electrons move from the higher concentration reservoir into your body. A paper published in the Journal of Alternative and Complementary Medicine in 2013 by researchers including Gaetan Chevalier measured red blood cell zeta potential before and after grounding. Zeta potential is the electrostatic charge on the surface of a cell in suspension. It determines the magnitude of electrostatic repulsion between cells. Higher zeta potential means cells repel each other more strongly and resist clumping. The study found that grounding through direct contact with the earth or a grounded conductor increased zeta potential on red blood cells, producing a measurable decrease in blood viscosity. Thinner blood, better flow, reduced tendency for cells to aggregate into rouleau formations associated with inflammatory states. The effect was measurable within 40 minutes of grounding contact. The electron transfer mechanism is physically coherent, and the zeta potential measurement is real. Whether touching a tree specifically produces clinically significant grounding effects compared to direct soil contact is less clearly established. The tree is a conductor, but the conductivity of the pathway depends on bark moisture content and wood species. Treat the grounding mechanism as physically plausible, experimentally supported in principle, and not yet fully characterized in the tree-specific context. That is the honest shelf it sits on. There is a third mechanism, and this one operates not through chemistry or charge, but through your autonomic nervous system, the division of your neural architecture that regulates heart rate, respiration, digestion, and immune function without any conscious input from you. Your vagus nerve, the 10th cranial nerve and the largest component of your parasympathetic nervous system, runs from your brainstem down through your neck, branches through your thoracic cavity, touching the heart and lungs, descends into the abdomen, reaching the stomach, liver, kidneys, and intestines. It is the primary highway of what physiologists call the rest and digest state, the counterpart to the sympathetic fight or flight activation you already encountered in the catecholamine data. Visual processing in your brain does not happen in isolation from autonomic regulation. Research in environmental psychology, particularly work conducted at the University of Michigan by Mark Berman and colleagues in studies published between 2008 and 2012, documented what they termed attention restoration theory. Directed attention, the focused, effortful kind you use to read a screen, solve a problem, or navigate traffic, depletes a specific cognitive resource associated with prefrontal cortical activity. Natural environments, and specifically visual fields dominated by fractal patterns, branching geometries, irregular organic forms, the visual signature of trees, restore that resource at measurably faster rates than urban visual environments. Brain imaging data showed reduced activity in the subgenual prefrontal cortex, a region associated with rumination and self-referential negative thought after nature walks compared to urban walks. Reduced rumination correlates with reduced sympathetic tone and increased vagal activity. The parasympathetic pathway opens. Heart rate variability, the beat-to-beat variation in your pulse that cardiologists use as a proxy measure for vagal tone and cardiovascular resilience, increases. The visual geometry of a tree, its branching structure, its irregular canopy, the fractal self-similarity of twig to branch to limb to trunk, is triggering a measurable shift in your autonomic nervous system through your visual cortex. You do not have to touch the tree. You do not have to smell it. Looking at it is already operating a mechanism. That mechanism is strongly evidenced at the level of behavioral and imaging data. The specific neural pathway from visual fractal processing to vagal activation is well characterized in outline and incompletely mapped at the synaptic level. Now step back from the tree itself and think about what the tree actually is. This is not a philosophical question. It is a physics and chemistry question with one of the most counterintuitive answers in the history of science. Put your hand back on that wooden surface. A mature hardwood tree, an oak or beech of 200 years, contains roughly 4,500 to 5,000 kilograms of dry mass. Conservation of mass requires that every atom in that wood came from somewhere. You could be forgiven for assuming it came from the soil. Roots penetrate meters deep. They extract minerals and water. The tree clearly uses the soil. Jan Baptist van Helmont, a Belgian chemist of the early 17th century, designed a landmark experiment to test this assumption. He dried 90 kilograms of soil precisely, potted a willow sapling, watered it only with rain and distilled water, covered the pot to exclude airborne contamination, and returned 5 years later. The willow had added 74 kilograms of mass. The soil had lost less than 90 grams. 74 kilograms of solid organic matter had appeared from somewhere that was not soil. Van Helmont concluded the mass came from water. It was the only substance he had added. The logic was clean and completely wrong. Water is H2O, hydrogen and oxygen, but wood is predominantly carbon. Dry wood is approximately 50% carbon by mass, with the carbon atoms forming the backbone of every cellulose and lignin molecule in the structure. Carbon is not in water. Carbon was not in Van Helmont's soil in meaningful quantity. The mass he was measuring did not have a visible source, and the answer was surrounding him at a concentration of 0.04% by volume, invisible, tasteless, odorless, and present in every cubic centimeter of air above his experiment for 5 consecutive years. Carbon dioxide, CO2. Trees pull carbon dioxide through stomata, microscopic pores distributed across the surfaces of their leaves, at densities of 100 to 300 per square millimeter. Inside the leaf's mesophyll cells, within organelles called chloroplasts, the carbon atom is extracted from the CO2 molecule and assembled into glucose through a process that requires energy. That energy comes from photons. Sunlight arriving at the leaf surface at 3 times 10 to the 8th power meters per second from a source 150 million kilometers distant carries energy in discrete packets called photons. When a photon of wavelength between 400 and 700 nanometers, the visible spectrum, strikes a chlorophyll molecule embedded in the thylakoid membrane of the chloroplast, it excites an electron to a higher energy orbital. That electron enters an electron transport chain, a sequence of protein complexes in the thylakoid membrane, and its energy is extracted in steps to synthesize adenosine triphosphate, ATP, and nicotinamide adenine dinucleotide phosphate, NADPH. These two molecules carry the captured solar energy forward into what biochemists call the Calvin cycle, a sequence of enzyme-catalyzed reactions in the stroma of the chloroplast where CO2 is fixed into organic molecules. Carbon dioxide is among the most chemically stable small molecules that exist. The carbon-oxygen double bond requires significant energy to break. The ATP and NADPH from the light reactions provide exactly that energy. Carbon is freed from oxygen, joined to other carbon atoms, and the resulting 6-carbon sugar, glucose, C6H12O6, is the precursor for every structural molecule in the tree. Glucose polymerizes into cellulose, chains of thousands of glucose units linked by glycosidic bonds, forming the fibrous scaffold of every plant cell wall. Cellulose is then interpenetrated by lignin, a three-dimensional polymer of phenylpropanoid units that fills the spaces between cellulose fibers and provides compressive strength and chemical resistance. Lignin is what makes wood hard. Lignin is what makes wood resistant to biological decomposition, and both cellulose and lignin are predominantly carbon, assembled from atoms that arrived as invisible gas. The wood under your hand is solidified atmosphere. Every gram of it was once CO2 drifting through the air, captured by a pigment molecule, broken apart by energy that originated as nuclear fusion in a star, and forged into solid matter by enzyme chemistry running in a cell smaller than the period at the end of this sentence. Van Helmont named the word gas. He derived it from the Greek chaos to describe the invisible vapors he kept encountering in his experiments, and he is credited as the first scientist to distinguish carbon dioxide as a substance distinct from ordinary air. He invented both the concept and the vocabulary for the very thing that was building his willow tree from beneath his nose for 5 years, and he never saw it. The man who defined gas did not recognize that gas was the answer. Now consider the structure beneath your feet. When you stand near a tree, the physical system you are in contact with extends downward through the soil in a network that changes the definition of what an individual organism even means. In the early 1990s, a forest ecologist named Suzanne Simard was working in British Columbia investigating why replanted clear-cut forests were failing. Douglas fir seedlings planted in cleared soil died at high rates, despite having adequate sunlight, water, and mineral nutrition. Simard hypothesized that the seedlings were severed from an underground network that natural forests depended on for resource redistribution. Using radioactive isotope tracers, carbon-13 and carbon-14, she injected labeled CO2 into sealed bags around individual trees, allowed the trees to photosynthesize the labeled carbon into their tissues, and then tested neighboring trees that had received no tracer. The Geiger counter registered radioactive carbon inside trees that had never been exposed to any labeled gas. The isotope signature excluded soil-water transport as the pathway. The carbon was traveling through a physical biological network connecting root systems directly. Mycorrhizal networks, the filamentous threads of fungi that colonize tree root tips and extend outward through the soil, their hyphae ranging from 2 to 7 micrometers in diameter, thinner than a human hair by a factor of 10, forming a continuous living fiber connecting the root systems of neighboring trees. The density of these networks in healthy forest soil is remarkable. 1 teaspoon of undisturbed forest soil contains kilometers of fungal filaments. Simard's work, published in Nature in 1997, demonstrated bidirectional carbon transfer between paper birch and Douglas fir through the mycorrhizal network. Subsequent research confirmed that the network topology follows the architecture of a scale-free network, a mathematical structure characterized by a few highly connected hub nodes and many lightly connected peripheral nodes, the same topology found in neural networks, the internet, and air traffic routing. The hub nodes are the oldest, largest trees. Simard called them mother trees. Kevin Byler, one of her graduate students, used molecular analysis to map the physical connections within a Douglas fir stand and found that virtually every tree was networked to the same continuous web. The traffic through this network is not metabolically trivial. Carbon flows from birch to fir in summer, when young firs are shaded and carbon limited, and back from fir to birch in winter, when birch has shed its leaves. The network equilibrates resource imbalances between connected nodes along concentration gradients. The same diffusion physics governing the Phytoncide cloud in the air above, now operating in the opposite direction, underground, through biological fiber. Beyond carbon, the network transmits chemical defense signals. When a Douglas fir sustains insect damage, it releases defense-related compounds into the mycorrhizal network. Neighboring trees begin producing defense enzymes before the insects reach them, a propagating chemical signal moving through fungal fiber in the dark below the soil. Samard's team also found that mother trees allocated carbon preferentially to genetically related seedlings over unrelated ones growing at equivalent distances. Identification of kin through chemical signaling in a network that has no nervous system, no brain, no centralized processing architecture. The scale of resource transfer and the significance of signaling continue to be debated in the literature. Some researchers have published critiques arguing that popular accounts overstate the cooperative framing and underplay competitive dynamics. That debate is ongoing and legitimate. The core experimental finding, directional carbon transfer through mycorrhizal networks, confirmed by isotope tracing, is solid. The tree you are touching is a node in a distributed system that has been operating continuously below the ground for longer than you have been alive. Now take that fact and hold it alongside the biology of the tree itself. Your cells age. Every division of a somatic cell in your body shortens the telomeres, repetitive DNA sequences capping the ends of your chromosomes, by 50 to 200 base pairs. Telomere shortening is the molecular clock of cellular aging. When telomeres reach critically short lengths, the cell enters senescence or apoptosis. Replicative capacity is finite. Your cells know how old they are because physics is measuring it for them in base pairs lost per division. Tree meristematic cells, the undifferentiated dividing cells at root tips and shoot apices, do not show this pattern. The cambium, the thin cylinder of meristematic tissue just inside the bark that produces new xylem inward and new phloem outward each growing season, divides without telomere degradation at the rates observed in animal somatic cells. Telomerase, the enzyme that rebuilds shortened telomeres, shows higher activity in aged trees than in young ones. Research examining ancient bristlecone pines found no molecular signature of cellular senescence correlated with organismal age. A 4,000-year-old tree's cambial cells do not behave like old cells. They behave like cells that have been given continuous renewal instructions. The tree does not have a biological expiration mechanism. It is not aging in the way you are aging. What kills trees is not their biology. It is physics operating on their structure. Wind force scales with surface area. A tree twice as tall presents 4 times the surface area to lateral wind load and amplifies that load through the mechanical lever arm of the entire crown acting at height. The bending moment at the base increases non-linearly with height. Water transport through the xylem, the dead hollow cells that conduct water from roots to canopy, purely through the cohesion tension mechanism, water molecules gripping each other in an unbroken column pulled upward by evaporation at the leaf surface, operates under increasingly extreme negative pressure as height increases. At approximately 100 meters, the negative pressure in the xylem approaches the cavitation threshold, the point at which dissolved gases nucleate into bubbles, the water column fractures, and transport fails. This is why the tallest coast redwoods, at approximately 115 meters, are near the theoretical hydraulic maximum. Not because cell biology cannot produce a taller tree, because the physics of water under tension will not sustain one. The square cube law operates on the trunk simultaneously. Mass scales with the cube of linear dimensions. Cross-sectional strength of the trunk scales with the square. Every increment of growth makes the structure proportionally heavier relative to its own structural capacity. Insects exploit wounds. Lightning preferentially strikes the tallest conductor. Drought fractures water columns. Fire sweeps the accumulated fuel load. The physical structure fails, while the biology inside it was prepared to continue indefinitely. The bristlecone pine, Pinus longeva, solved this by minimizing its structural exposure entirely. Growing at altitudes above 3,000 meters in the White Mountains of California, on dolomite soils hostile to competing vegetation, in an atmosphere too dry and cold to support most pathogens, it grows less than a centimeter of new wood per year. It uses sectored architecture, keeping only a narrow strip of living cambium connected to a fraction of the root system and a handful of needle-bearing branches, while 90% of the wood is dead and resin-saturated, resistant to rot, erosion sculpted rather than decay consumed. Its needles persist on the branches for 40 years, eliminating the metabolic cost of annual leaf replacement. It has traded scale and speed for structural longevity. The oldest confirmed living individual, a bristlecone called Methuselah growing in the White Mountains, is 4,857 years old. It was photosynthesizing when the first stones of the Great Pyramid were being laid at Giza. Its precise location is withheld by the United States Forest Service. There was an older one. In 1964, a geography student named Donald Curry was coring bristlecone pines in Nevada for dendrochronological climate data. His increment borer jammed in one particular trunk and he could not retrieve it. He obtained Forest Service permission to fell the tree. He cut it down and began counting rings. The count passed 1,000, passed 2,000, passed 3,000, passed 4,000. The tree he had just felled was 4,862 years old, the oldest known living organism on Earth at the time, destroyed because a sampling tool became lodged. Curry rarely discussed the event publicly for the remainder of his career. That tree had been accumulating starlight into carbon bonds since before the wheel, before writing, before any continuous human institution that still exists today, and it ended not from any failure of its own biology, but because a human being needed to retrieve a metal tube. Go back to your hand on the wooden surface. You have been there, if you followed the opening instruction, long enough now to have experienced the thermal illusion of warmth that low conductivity produces in your somatosensory cortex. In that same interval, you have been breathing air containing phytoncide molecules moving along a concentration gradient from the wood surface through the gas phase into your alveoli and bloodstream, beginning a process that research documents will measurably elevate your natural killer cell surveillance for the next 7 days. 7 days. Not from a pharmaceutical course, not from a clinical intervention, from molecules following a physics gradient from wood to lung. If the surface you contacted was a living tree in soil, electron flow through a grounded biological conductor was modifying the zeta potential of your red blood cells and altering blood viscosity in a direction associated with reduced inflammatory aggregation. The visual cortex processing the branching fractal geometry of the tree was reducing subgenual prefrontal activity, lowering rumination, increasing vagal tone, shifting your autonomic nervous system toward parasympathetic dominance. And beneath the soil, a mycorrhizal network connecting this tree to the others around it was redistributing carbon along concentration gradients and propagating chemical defense signals through fungal fiber at a depth where no light reaches and no sound travels. The tree you are touching is not a single organism. It is a node in a distributed living system running on the same diffusion physics as your own bloodstream. Its wood is atmospheric carbon assembled by photon energy from a nuclear reaction 150 million kilometers away. Its cells carry no biological clock. It is not aging. The physics of its own structure will eventually destroy it, but its biology would have continued. You have a clock in every cell. It is counting down in base pairs. The tree does not. Touch it. You already have.